Hi, Will. Nice to see you. Thank you for joining today.
Good morning, Natalie. Great to be here. Thanks for the opportunity to talk some with you guys.
I'm really glad you could. You have such an interesting career path. I know a little bit about it, but certainly not all about it. And I welcome the chance to hear more. I know that you're in a really interesting spot right now. Do you want to maybe start by saying what your job is now, and we'll back up from there?
Yep, sure. That sounds good. So right now I'm a scientist at Corteva Agrisciences, which is an agricultural biotechnology company that's headquartered in the United States. And so we provide all kinds of services and products in the agricultural space, for a very large portion of food production in the country and the world. And there's all kinds of technologies that go into that, with protecting crops from insects, improving their yield through breeding, genetic engineering technology with transgenic traits. So it's a pretty broad area.
It sounds great. Is that your lab behind you?
No, this is just a picture from an early career inspiration for me. This is a movie, Jurassic Park. The first one came out when I was in elementary school, I think, and it kind of got me thinking about working in industry when I was a kid. So
that's so funny. So it wasn't a cautionary tale for you. It was an attraction. So, but I'm guessing your work is actually a little bit different than all that. Like, do you work with a large group where you are currently working? Is it a bunch of people working together on a project?
Yeah. So I work in very early phase discovery research. So basically, at the very beginning, where we're thinking about new ideas, new technologies that we can use, to make the crops grow better and provide, better products for farmers. Because I'm so early in the process, exactly what we're doing, it's kind of a proprietary thing, because we normally we wait until we've really shown it works and patent some of the inventions before we publicize a lot of it. But I work protection space, so trying to identify ways we can genetically modify the crops to protect them from insect feeding. And this is something that's already, there are versions of this that are already out and have been out for a very long time. And we're trying to make the next generation of that technology that works better than what's out currently.
It sounds really important. Protecting and enhancing our food supply is such an important effort now, with just climate and population and all the other demands that are growing in our environment. So it sounds like an important job. Are you leading a group or part of a group, or do you move around from group to group?
So when you're in industry, there's multiple ways that you can be a contributor, and there's all different levels that you can contribute. Basically amounts of training that you can have before you can come in. And so I'm at a research scientist level, so I do a lot of strategy. I'm project leaders, things like that. But I've kind of chosen, or the path I've been down has been still staying as more like an individual contributor or scientist. So I don't have a whole team that I manage from like an HR standpoint, but I have teams that have large numbers of people that have to collaborate together on a project. So if it was Jurassic Park, I would be in charge of maybe coming up with Stegosaurus or something like that, and then there would be people from a variety of teams, that would have like their boss or something would be from different places, but they would all collaborate together and work on the project, under kind of my direction. And so that's one way of doing it. There's many different management styles, but we have things kind of broken up with project management and then sort of people management within Corteva.
Yeah, I think it's so interesting because these are big questions, right? They're beyond what any single person could really fully master and make a lot of progress. And I think science is much more collaborative than people sort of envision it to be. But and that seems to hold true for industry as well as for academia, which can be surprising to some people, I guess. So.
Yeah, it's really, I think, I don't ever want to discourage someone, from, thinking like if they've got an idea that they can't drive it forward and make a lot of progress on their own. But, you're in this competitive world that we're in with companies and stuff. You very rarely see a company that only has one person. And even, a lot of people say in the venture capital world, they don't even really like to have one founder anymore. They like to see that, well, there's, a few people that are there that can share the work, or you have different complementary skill sets. And so, again, in the world that I'm in, I have a lot of background training in molecular science, bioinformatics, and chemistry, biochemistry, things like that. But I don't have that training, in agronomy or plant health or entomology. And so, being able to have members on the team that have those skills really enables, the ability to work on some of these complex problems.
I think that sounds great. That actually brings up an important question, which is about your training. A lot of young people are thinking, what is the most important thing I should be doing or learning in order to get to be a practicing scientist or a practicing engineer? Sounds like you came at it from a molecular science side. Where did you go to school, and what did you study?
So, I'll take it all the way back early on to the beginning. So, in high school, I took courses with dual enrollment at a community college, and then I also took some AP courses. I was involved in, some clubs, student clubs, things like that. And I had in my head, say when I was in high school, I grew up in Southwest Florida, and kind of I saw you had Irene on. She was talking about growing from a farming environment or something. I wasn't in a farming environment, but I was in a place where there was no major university. There was no, professors around or startup companies or anywhere where I could get any exposure to genetic engineering, except for maybe going to the library and checking out a book, like a physical book, for anyone out there who, knows that those still exist. But they, that you have to go there and it might be checked out and you couldn't get it. And so I learned a lot through taking some of the fundamental science courses. So I was very interested in physics, math, chemistry, calculus, computers were really starting to take off, the personal computer, when I was younger. And so originally I wanted to be a computer engineer. I was very, very much like, oh, well, I want to do computer engineering. And I looked up at, I was in Florida, Southwest Florida. So I looked up online what the curriculum was. And I said, well, how many of those courses can I take at my local community college? And I started taking, calculus 2, chemistry 1, chemistry 2, physics, all these different things. And, it was helpful. But then I got into the university, and it's time, they're very much, and I don't know how much they still do this now, but, it's very much you show up on day one and they say, well, what's your major going to be? What's your career going to be? What do you want to be when you get done with this? What are you going to do after you graduate? And you're, at least to me, maybe to other people, you're saying, well, I just got here, I just got here. But so I got in there and I said, well, I'll be a chemistry major. And, I thought for a while, for a very short period of time, that maybe I wanted to be a doctor or a medical thing, which of course is very strange because I had just been doing all these courses to be a computer engineer. But you get these social pressures and things of like everyone, a lot of other, if you see a lot of your other friends or people or parents are saying, well, oh, this is a good career for you to go into, you can make a lot of money, or, it's difficult, or you can still do science and things like that. And so for a short period of time, I was pre-med, kind of just by, sort of being pushed in that direction. And I quickly found, I was pre-med chemistry, and I kind of found that was unsatisfying, because to meet the requirement to be pre-med didn't require taking the full-throated scientific courses like physics with calculus or, the more advanced chemistry courses. There was just a set of prerequisites that allowed you to take an exam, and then you did well on this exam, and then you could, go forward. And so I went from chemistry to being chemical engineer, and then I got to a point where it was the midway through, I think it was my sophomore, I started my sophomore year, and I had a choice to make that I could either take this course called Energy and Mass Balance, which is for anyone out there who's a chemical engineer, this is like, this kind of locks you into the path a little bit. It's a kind of long course about thermodynamics and engineering and chemical engineering. It was a 5-hour course per week. And, or I could do bioorganic chemistry with this one professor that I really liked. And I was like, well, I really like this. I really got interested. I was very interested in this topic because I had taken organic chemistry with the professor. And it was like, yeah, you know what, I'm just going to go with what I like. And so I went down that path. The professor I formed a good relationship with, and he asked me to do undergraduate research with him. So I got, after my sophomore year, doing undergraduate mechanistic enzymology, which I really thought was interesting and fascinating. It became a kind of a good merge of the different skills that I had kind of picked up with, I had the interest in computers, and so there's a lot of modeling. This is, again, for people who are maybe a little further along, you can represent molecules, proteins as 3D structures that you can kind of interact with, almost like it's a video game, and get insights into. So I was able to do that, was able to do some kinetic modeling. The genetic engineering component, doing site-directed mutagenesis was really, kind of, in some ways it was very surreal, for me personally. I remember when I first was explaining to me the research project, and I said that it can't possibly work that way, right? It's like, we have this, like you're saying, there's a tunnel through this enzyme that a substrate, channels through from one active site to another. And I said, how can that possibly work? And he said, well, your project's going to be to try to block the tunnel by, doing mutagenesis on, some amino acid that lines it. So there I am looking at a 3D model on a computer, trying to decide what I want to do to try to block this tunnel to then do a kinetic experiment. And I thought it was great. And, I got working with him, I got the chance to go to conferences. I went to an ACS conference in Atlanta, and I presented a poster. I even got the chance to go to, I got to go to Como, Italy, Lake Como in Italy. It's like something in, there was a Enzymology International Conference, and I got to go there and present it. And that was my first time to be outside the United States. And this is like I think like Ocean's 11 or something was like partially filmed there. And so, this is like a pretty, exotic place. And,
yeah,
it was pretty neat. And so then so after that, I was like, well, I really like research. I was fortunate to have some of the work I did in a peer-reviewed publication, which was exciting. Because that's another thing that, I think it's starting to change a lot now too, for anyone that's a young person that's out there. There's, the currency of being in academia has always been, and maybe it still is, the paper, but I do sense there's starting to be this transition to more self-publishing or sharing things in social forums or on YouTube or on Twitter, or these things where, I'm me as a scientist, oftentimes if I'm trying to find what's the most cutting edge interpretation of something, I'm actually looking to certain individuals on social platforms, discussing it kind of in the open, which never happened before. You used to have to be, at like a social hour at a conference to get any insight into some of that stuff. So it's kind of a neat time. So then I went to graduate school. I got into Berkeley out in California. And one thing I'd point out, I'd say too, from a career standpoint, when I was in high school, I never really cared too much about, much of anything, really. I was just kind of careening along through life, and I was like, well, I like this, I like that. And I actually wasn't able to even be in the dorm my freshman year because I forgot to send in any forms related to staying in the dorm. And I only applied to one university for undergraduate, that's terrible advice, by the way. I suggest that you don't do that, that you at least could consider multiple options, because I only applied to one place. Luckily, I got in, forgot to fill in the dorm. And then it was like the total inverse of that for graduate school. For graduate school, I was like, here's all these good schools. I'm going to assess all the different faculty members there. What are they ranked? What are the, who do I want to work with? How am I going to connect with them? Like it was like a total 180. And, so then you go out, and you visit all these different places. And, I had a very embarrassing moment in hindsight, which was, I was kind of down between a few places, and there was one place, I won't say exactly where it was, but it was in the Northeast. And I had gone there during a blizzard, and I had no idea what it was like to be cold because I had grown up in Florida. And so there was like feet of snow. And while I was there, me and some other person, we had gone to the mall and bought jackets. And I was just like, I told the person on the phone, I said, look, I really like your program, but it's just too cold there. I just can't handle, I don't think I could handle being in such a cold place. And, to the credit of the professor or the person, they told me, and this is good advice. They said, that's a terrible reason. Well, that's a horrible reason to decide where you're going to go to graduate school. And I was like, all right, fine. I just like the people at Berkeley better, and I just like their program better. And then the person said, well, that's okay, And what was funny is they told that story to people who came in subsequent years. That there was some person that came that
so now you're a living legend.
Well, I mean, or the butt of a joke or something. But it was funny. Yeah, it was funny to hear from my friend, one of my friends who had been an undergrad at Berkeley, that when I was first starting there, that went to that same place and was told about oh, there's some person who didn't know what it was like to be cold. And so anyways, I was at Berkeley. I got to work in a very, I got very passionate about the energy sector and things like that. Like today I think it's become, there's so many young people that are very passionate about the environment, especially with concern with climate change and the plastic in the ocean, or any sort of environmental type of thing. There's way more awareness and concern now than I think when I was a little bit younger. Or at least there wasn't much where I grew up. It wasn't a very big topic. And but I was into it for some reason. Like I made some decision. I was like, well, look, I don't want to do biomedical because I don't really, me personally, I just didn't like the idea of being in that medical environment or doing research on, humans or living animals or things like that. For me, it didn't personally like that as much. And it was to work in this new sector, which they were calling it bioenergy at the time, or the bioeconomy. And this is kind of around where some of the ideas of the synthetic biology kind of really blew up and took off. There was a lot of funding for some areas of research that had not really been funded much before. Most funding went to medical applications from the government agencies. And so that we had a big investment into renewable energy and bioenergy. And a lot of people started to think about and work on it and work in some of these areas. And so I got involved with that. And I was very interested in, fuel production, as well as conversion of one thing into another. So my project was really focused on how you could take the fiber portions of plant matter, which is currently unused, and convert it into the equivalent of sugar or sucrose or glucose. And so this was a ton of people were working on it, and because it was one of the principal problems. So for anyone viewing here who doesn't have a car yet or hasn't looked into this that much, you know, gasoline has additives put into it, and one of them is ethanol. And there's a limit of up to 15% or so, 10 or 15%, depending where you live, of ethanol that's added to your gasoline. So that's actually a huge portion of production, you know, in absolute terms. And if you live in the Midwest and drive around, you'll see these giant facilities, which are fermentation facilities, where they are producing alcohol, ethanol, and this goes into many products. So it's not only into all right, sorry, yeah, I'm back. I had a technical difficulty with my internet connection. So I was saying that it was in graduate school, we're working on this bioenergy problem. At the time, the gasoline price was very, very high, like $5. It was going up to $5 a gallon. You had the environmental concern, you had the national security concern that we're running out of it. You had the price getting so high. It was really kind of a wild time. And so, you know, you felt like you're working on the Manhattan Project or saving the world. And so working on this really hard, thinking about it. My PhD was on what I thought was the most important, difficult part of that problem, which was this depolymerization problem. And so it was very exciting. I really liked it. I was fortunate enough to work with some other really good people. Had some good advisors. A lot of opportunities. I got a fellowship also from National Science Foundation, which made it, you know, it was even more supported from a financial standpoint. And independence. It gave me some independence from not needing to be kind of on a specific grant per se. And so it was great. And was able to learn a lot about fungi, filamentous fungi, which are, you know, you can learn about things on them on YouTube, but they're very important in the industrial biotechnology sector, not yeast, but the filamentous kinds like mold. And people are making proteins out of them, enzymes, all kinds, even, you know, food, turning them into food in some cases. So it was very exciting, and we found some fundamental mechanistic enzymology things that was really great about oxidative enzymes that are involved in depolymerizing, kind of cracking the nut of the plant cell material, plant cell walls. And then so, we also
the chemistry and the chemical engineering all sort of continues to fit into this biological framework, and now you've taken it from, you know, enzyme site-directed mutagenesis to plant sort of biomass conversion, right?
Yes, yeah, it was yeah, and it was such a from a you know, for anyone aspiring chemical engineers out there, it was such an interesting area because in traditional enzymology, you basically are always kind of assuming you're under some sort of diffusion limited environment, that, you know, substrates in aqueous solution are kind of bouncing around in a diffusion limited fashion, and then they come into contact with your catalyst, which is the enzyme. Whereas in the world of I guess heterogeneous catalysis, which this is, and it hadn't been studied very much from an enzymologist standpoint, because quite frankly, the experiments are very difficult to do. And so, you know, our substrate was always insoluble. It was poorly defined how much of it was available. We couldn't synthesize standards of it because, despite the fact that things like cellulose or hemicellulose or oligosaccharides are all over the place, you know, it's in, you know, boatloads of it are in your oatmeal or, you know, in corn flakes or whatever, to get them in a defined, purified form that you can do careful experimentation on, like chemists and biochemists do in the medical space, was basically impossible. It was practically impossible. It was you know, you would have to spend thousands and thousands and thousands of dollars for milligrams of these oligosaccharides, and then they may not even work because the enzyme actually recognizes a crystalline surface or something. Right, and so it was, the science of it was really interesting. And then the reaction, it turned out that it was a new type of inorganic biocatalyst. So it was like and that was, you know, I think one of my advisors, Michael Marletta, a pretty well-known mechanistic enzymologist, made big contributions, or, you know, throughout his career into, like, you know, very serious enzymology things. I think he was truly startled the day that I told him. I said, I think the thing that's really important for this process is a novel, monooxygenase that has copper in it, and is working mechanistically in a way that we've never seen anything work before. And I think he was in kind of a state of disbelief maybe initially, and then was like, wow, this is pretty cool. This is pretty cool. My friends at the bioinorganic Gordon conferences will find this interesting. They're not going to just say, oh, you're doing it, because there's a lot of poo-pooing, I think, rightfully so, that you're just working on a hydrolysis mechanism, right? Which is the thing that undergraduates will learn, general acid catalysis or something, right? And it's a very as opposed to some serious electron pushing and different oxidation states and all of this. And so it was neat. I also got a lot. I brought back my knowledge of computer, or sort of my love of computers and using computer programs in graduate school, because it was right in this time. I was in graduate school from 2006 to the beginning of 2012, fall of 2006 to beginning of 2012, and this was around the time that the sequencing revolution, the next generation sequencing revolution, was taking place. And so we were going from a position where we had essentially no genomic resources for any fungi outside of like yeast and maybe one or two other ones, to where it became feasible that we could do it ourselves in the lab. And so that this required some computer programming, these DNA assembly algorithms that were applicable to these technologies were just being written. And it was very exciting, because I remember I was working with some stuff that was very cutting edge, doing de novo transcriptome on some of these fungi that I was working on. And it was a great environment. I remember I talked to our IT person. I said, hey, I need a powerful computer so that I can, so that I can try to do some of this stuff, do this assembly. And he's like, all right, well, let's go build it. Let's build it or find it or buy it. And, we built it, we installed Linux, I got these programs running on it. And then it was very eye-opening. It was more like, for me, it wasn't just that it was like a video game where I was looking at, some model in 3D, but it was more like I can use this data in a way that I never thought possible to make predictions about the experiments that we're going to do. And so I had a paper related to systems biology, integrating a lot of different types of data to help make better decisions and predictions about how these things work. And it was it was very neat, and it's very applicable today. It's a very if again, from a career advice standpoint or a training standpoint for anybody interested, I think it's really important early on, get some foundation, know some foundation in math and physics and chemistry. And then, once you feel like you've got that intuitive understanding of the basics of those, then just kind of, make sure you know something about the computer too. Of how can you use the computer to take in data that's more than you can handle, to read yourself. And if you can do that, then just kind of follow your interests, I think. And so that helped me out. That helped me out a lot. And I use it all the time in my current work now. I don't do a lot of electron pushing in my current work right now, but I do a lot of data integration and experiment interpretation. So
as I'm listening to your path, it really does seem to fall into that pattern, right, of using foundational science and courses and learnings to set up your intuition about things and your basic framework for thinking about the projects, and then using computational tools when you have to go beyond intuition, and then following things that make you, that you find genuinely interesting. It seems like you have been driven by curiosity, and it has taken you into places that would not have necessarily been front of mind when you started, but that sure seem to pan out very, very well.
Yeah, I'm happy with, overall how things have turned out. It's been, there's, I tell people all the time too, you're going to run into bumps in the road. You're going to have moments where you're like, what am I doing? I need to pick. I've got to pick, like, oh, I'm going to do this, or I'm going to do that, or, I've got to get an A on this test or whatever. Right. And, maybe in the moment that's really the case, but your whole trajectory, your whole career is the integrate, just like I'm saying, oh, I'm integrating a little knowledge of physics and computers and biology. It's the same way with your career, or how you develop your skills. It's that yeah, and so because there was definitely some time, like I'll tell you, my project I'm talking about with graduate school, where I said, oh, it's so successful, I've discovered some really great things. There was a moment where I went into my advisor and I said to him, I said, look, it's time that we publish to tell the whole world that this doesn't work. It just doesn't work. And he said, he said to me, he said, well, people don't publish papers like that very often. I said, but we've done so much and it just doesn't work. And that was, and that was right before the key breakthrough, right, that, where it finally came together. And, but yeah, it was so. Yeah, it was fun. I do always want to point that out, because whenever we tell stories, we normally tell them like we create a story that says like, oh, I did this fun thing and then that fun thing and then this great thing and I linked it all together. And while it was happening, it certainly didn't seem so directed like that.
I think that is so true. We have these narratives. Once we reach the end, we look back and we think, oh, that's where all those pieces fell in. But in the moment, in the process, it sure feels like the direction and the end point aren't always as clear as as the stories we tell make them seem to be, right?
Yeah, definitely. And you can always, I tell people too, you can always update your story if you come up with a better one, down the road, yeah, you can always update it and be like, I realized another thing that I left out that was really important, or emphasize or de-emphasize. So it's but for me right now, I say it's really important, if you're, I think, again, if you're a young person right now, use your computer, and, follow people that are doing work that you're interested in, try to get a good core set of skills, find people in your local community where you can interact with and talk about ideas that you have, or practice your skills, join clubs at your school, participate BioBuilder, fill your day up with things that you like, that you want to get better at. And and I think that normally that can work out pretty well. That could work out pretty well, because things are changing a lot. I can't be prescriptive and say that what I did specifically is the right thing to do today, but because the world is so different now. But yeah, I think it's generally pretty good to say, pursue what you like, what you're good at, and find some other people that you think are even better than you are right now, and see if they'll help teach you what they know.
I think that is great advice. And I am so grateful that you contribute to the BioBuilder community and share your wisdom and advice to help exactly those individuals that you mentioned, the folks that are interested in computers and biology and math and engineering and making the world a better place, environmental issues, all those things, and trying to find a way to bring it together. Within BioBuilder, we try to do that, and we very much rely on the wisdom of people who have traveled that path already, like you, to turn around and help the next ones along. So thank you for all that. And thank you for taking the time today to share your story. It's such an interesting one.